Hello, everyone. Welcome to the fourth MACSMN Mathematics and Computer Science for Materials Innovation. Our next speaker is Mihaly Varadi from EMBL-EBI. And Mihaly will talk about navigating the protein data universe. Over to you, Mihaly. You're muted at the moment.
Yes, sorry, my control panel was playing hide and seek. So thank you very much for the invitation and the introduction as well. I regret not being able to be there in person, but I hope to give you an overview of different data resources and different types of data that are very useful for structural biology and using structural data to answer questions, scientific questions. I will start with my main messages. And in the protein data bank, so I work in the protein data bank in Europe team, and in the protein data bank, we have about 200,000 experimentally determined structures for proteins and nucleic acids. And that's not a lot, to be honest, if you consider different data sizes at different domains. However, there are over 800 million predicted structures, and these are mainly from databases like the AlphaFold database and the ESM metagenome atlas, which you could use as very powerful tools to tackle problems that were previously very hard to work with. Mainly, these are in drug discovery or understanding disease mechanisms, but also many other areas where these structures can be useful. However, doesn't matter if it's coming from experimental methods or from computational predictions. Just knowing the XYZ coordinates of atoms is often insufficient to answer specific scientific questions. You need to know more. You need to investigate these structures in their context. And that's what the second half of my presentation is going to talk about, which is this PDBe knowledge base, which is an open consortium where we aim to place these structures in their biological and biophysical context. Through fair access mechanisms. So fair in this context means findable, accessible, interoperable, and reusable. And basically this means that the data should be something that helps you, and it's easy to find, and it's easy to use and easy to reuse. So going forward, this is how I imagine the flow of this presentation. First, I will be talking about the background and kind of setting the scene, and then talk a bit more about the PDBe-KB resource and how you can use that and what type of things are we trying to do there now. So first of all, to set the scene, you may or may not know, but there's a really big gap between the known sequences, protein sequences, and the known protein structures. And this gap is increasing. So just in the past 10 years, it grew by 10 times. At the moment, just in the UniProt database, we have about 250 million protein sequences compared to the about 200,000 PDBe entries. And actually, those are for about 62,000 unique proteins, because in the PDBe, often you have more than one entry, more than one structure for the same protein. So it's a very tiny fraction of the UniProt database that's covered by experimentally determined structures. And this is just the UniProt database. It doesn't include metagenomics data and other types of data. So as I mentioned, this gap is just growing. It's not getting any better. And it's quite logical as well, because sequencing, that's very cheap now. It's kind of easy, especially compared to determining the structure of a protein, which is often a hit and miss, and often you spend years on trying to solve a more complicated structure. So on one hand, predicted structures could probably help and close this gap. And indeed, for over 50 years, it has been postulated that in theory and within certain limitations, if you know the amino acid sequence of a protein, you should be able to determine a structure for that protein. And indeed, for the past half century, researchers and software developers, they were trying to create increasingly sophisticated ways of taking the sequence, the amino acid sequence, and coming up with a structure that's a useful model in order for you to solve actual problems, biological problems. A few years ago, so in 2020, there were a couple of major breakthroughs in applying AI-based techniques to model prediction. So very briefly, just a quick overview of how this structure modeling evolved over time. The traditional way is, if you have a structure, if you have a protein and you're interested in coming up with a model for that, you would see if there are any sequences that are similar to your sequence of interest, and see if they have any structures in the protein data bank. And this works kind of well, because then you can take a template from the PDB or several templates and try to come up with a model based on that. But obviously the problem there is you need a template. And even though these template-based structures are often reliable, you often don't have a template, so it doesn't work. Another broader class of approaches is called ab initio modeling. And here often you take, you don't need a template structure, and often you rely on advanced force fields, similar to those used in molecular dynamics. And you try to start with kind of like a string and try to fold it up, try to have the force field folded up into a meaningful structure. The problem there is that it's computationally very expensive. And even today, for larger systems, it just doesn't work. You don't have the compute capacity often that you would need to run such a modeling. But then came this new generation of modeling tools, which are deep learning based. And in some sense, you could consider them as ab initio because they don't rely on templates. Often these AI models, they can choose to use the template, but they often don't use it even if the template is there. And you might have heard about several of these tools. There's AlphaFold, ESMFold, all kinds of folds, and they are very reliable now. And often they have results that are comparable to experimental methods, but they have very strong limitations as well. Well, clearly these new tools, they impacted many fields of life sciences. And one of the things that surprised us a bit, at least at the protein data bank, is in terms of structure determination. We saw quite a big jump in the number of deposited structures in the protein data bank, because now having a model helped many people who already had experimental data to explain the experimental data and to come up with a very good experimental based structure, but with the help of models. So that was an interesting use case that we saw, but it clearly affected teaching, training, many data providers like Uniprot, like ourselves, others as well. And of course, if you are doing bioinformatics and you work with structures, then it's kind of a new gold rush for you, because there are tons of data that you can take and analyze. And it also came up or came with many challenges as well, because it turns out that many of the algorithms that were developed and used in structural bioinformatics, they didn't really anticipate this amount of data. I mean, in the PDB, we had 200,000 structures. Anything that works within a reasonable amount of time for 200,000, that was good. But now you have millions, many millions of structures, and things just don't scale, it turned out. So one thing that's I think very important to note here is that all these new tools, so AlphaFold, ESMFold, all the others, they took advantage of data that researchers created over 50 years. And without the painstaking effort of all those people who toiled and tried to determine a structure or to sequence proteins, without that data, this would not be possible. So resources like Uniprot, PDB, those were really essential for these AI tools and to train these models. And now, in a sense, I think it's fitting that these tools are giving back to the community and the scientific community. And finally, these people can answer questions that they couldn't before because they just didn't have a good enough structure. So I think it's kind of a good synergy between this next generation of AI tools and the data and the people who created the data. And as I mentioned, some of the key areas that were impacted are structure determination, drug discovery, and functional analysis. Well, obviously, if you have the structure, you have a better chance of knowing what that protein does. Just to provide a few highlights that I really liked. These are from people who used AlphaFold and tried to solve some real problems. One of the problems that we are facing as well, humanity, is that bacteria are becoming ever more resistant to many of our antibiotics. And one of the things that bacteria can do is they use enzymes that are embedded in the membrane, and they modify their own membrane so that the drugs can't bind anymore. And these are, of course, membrane proteins, so they are embedded in the membrane. And why it's important is that they are notoriously hard to solve using experimental methods. And so in the PDB, you won't have too many of these. You won't see too many membrane proteins, or at least not the full view of these structures. And it turns out that these new tools like AlphaFold, they are quite good at predicting membrane proteins. And now this is a new tool that actual people in research laboratories working against antibiotic resistance, they can employ and or deploy and use it to solve these problems. Another example that I liked is about bees. And of course, bees are very important because they pollinate stuff for us, and otherwise we would be in quite deep problems. So the problem there is that honeybee populations are on a decline globally. And there are many reasons, but the major reasons are to do with certain infections and parasites coming. And so to help the bees and help ourselves as well, it is very important that we precisely understand how their immune system works, because then we can design drugs, or we can come up with strategies how to make them stronger and more immune to certain diseases and pathogens. So one of the key components there, one of the key proteins is called vitellogenin, and that turns out to be a very important protein in all the egg-laying species. However, especially for bee, we didn't know how this protein looks like. And here you see an image of that. So it's quite a big and complicated structure. And probably that's why people struggle to determine it using experimental methods. But now using these AI tools, it's much easier. And now you have a quite reliable structure that can really help. So just to step back a bit and just very briefly talk about AlphaFold. Of course, it's developed by Google DeepMind. And in 2020, it achieved very good accuracy during this critical assessment of structure prediction. This is something that happens every second year, where they compare different tools. And at that time, it was declared that it kind of solved the folding problem. Now, in retrospect, we know that it's not true. It's not really solving the problem because it doesn't know anything about folding. It knows, it can predict the actual protein fold, but not the process of folding. It doesn't understand anything about chemistry and how things happen. It just, it's very good at predicting the final state. But even there are some certain limitations that I will mention. It has some obvious strengths, like it works based on only amino acid sequence. It doesn't need a template. If there is a template, then it may or may not use the template. So the AI can choose that. In this case, I'm not going to rely on the structure in the PDB. And that also kind of makes sense because in the PDB, we do have some lower quality structures as well. And then in that case, it's quite good choice to discard those and not use them as templates. It has 3 independent outputs. And that's usually something I always try to explain, because you have to use all 3 of them if you want to do anything with these predicted structures. So it has the predicted atomic coordinates. It has something called PDBe-DT score, which is a local confidence metric, and another thing called predicted aligned error. The atomic coordinates, if you haven't worked with these type of things before, basically it's XYZ coordinates for atoms. And there are 2 formats. I just want to highlight here that the PDB format is, confusingly enough, not the official format of the PDB anymore. It's a legacy format that used to be the official format. Now it's obsolete. And in fact, it will start breaking around the end of this year, because we are running out of identifiers for small molecules, and there need to be a change. So there will be 5-letter codes for small molecules. And from that point, new entries will not be PDB compatible. So that's something to keep, to look out for. But we have the official format, which is the MMSIF data format. And it's a much better format. It's much less restrictive. And you can include all types of information in there. So the PDB format, that was very restrictive because it was, well, the PDB is over 50 years old now. And at the time when PDB format was designed, it was restricted by punch cards. So basically it had 80 columns because punch cards had 80 holes in it. And so it's very restrictive and it's very old format. Now, if you can, try to use the MMSIF format instead. The second output is this PDBe-DT score. So that's a local confidence matrix. It tells you what the AI model thinks about the accuracy of a certain residue in the model. And if you see an AlphaFold structure, chances are that they are colored something like this. This is a color coding that we came up with at the beginning of the work with DeepMind. And Blue things are good. So dark blue, that's usually comparable to experimentally determined structure accuracy. Then lighter blue is still very good, but then as you go lower, you need to look out. And actually there are some details there which I don't think I can cover now, but there are some things to look out for when you have low PIDDT. And the final thing that the final output I usually spend a bit of time to explain, because it's very important that people use this too, and they often neglect this. So it's called predicted aligned error, and it's a pairwise score. So for every residue pair in the structure, you have a score. And what this tells you is, if you look at the structure, how confident can you be that residue 1 and 2 are in fact positioned as you see it in the structure? So I will show you an example to correctly explain that, but basically it tells you, is it really next to each other? And so here are a few examples to try to explain. On the left-hand side, you see a domain, and you might wonder if this domain, well, or kind of like folded part, is it really next to it? So are they really next to each other? You might not wonder, because you might think that, okay, you look at the structure and this is what it looks like. This is the structure. And people often do that because they are used to things from the PDB, where you see a structure and that is the structure. With an AlphaFold structure or other AI-based structures, that's not the case. You actually have to be careful and investigate, are those things really next to each other, which is a bit confusing. So here, if you look at this green domain, and you wonder if the gray domain is next to each other. So if you select that, then on the PAE plot, it shows you this dark box. And when it's dark, that means that you can be confident that yes, those residues, those residue pairs are as they are appearing in the structure. If you select the other one, this other domain in the middle panel, then you see that it's still in the dark green box. So the whole thing is in a dark green box. That means that all the residue pairs, they are confidently oriented next to each other. But for example, if you look at this small helix here, and maybe you would say that, I see another helix there, and maybe this one there, and maybe I would imagine there's some kind of pocket that I could target with a drug molecule, or whatever. Maybe I will try to disrupt this interface. But actually, if you look at the PAE plot, it's isolated. It has nothing to do with the other regions. This means that this part could be anywhere. It happens to be there now in the model, but there is a flexible linker that could move all over the place. And these helices, they are not next to each other. They happen, it's kind of like a snapshot, but they could be anywhere in the structure. And that's very important. That's something that many people ignore when they work with these structures, but it's key to actually using these as useful tools. In terms of certain limitations that AlphaFold has, it was not trained to predict the structural effect of mutations. That's one of the things that people often ask, can I use it to understand what a certain mutation does to the structure? Is it destabilizing it, or what does it do? AlphaFold can't really say. There are other tools that can, like FoldX, where you can take AlphaFold structure and try to see what happens. It also can't put ligands in there. Some other tools, they try to remedy this problem, but AlphaFold is not plugging in ligand molecules into the structure. And it doesn't work on nucleic acid structures either. It only accepts the 20 standard amino acids. And one of the interesting points that I like to highlight is, even though by default it creates 5 models per run, they are very similar. So if you have a protein that has distinct conformations, which are different to each other, and these are biologically relevant, so it has a meaning why they are different, AlphaFold is not going to predict them. It always is going to predict one conformation and completely ignore the other one. Some people are trying to trick it, kind of like giving it less information, but then well, the output is also less certain, so you can't really rely on that. And this means in practice is that if you only have a sequence and you run AlphaFold, and you happen to investigate a protein that actually in nature has multiple distinct conformations, you don't really have any idea which conformation is it giving you. Here's an example to show that. It's a protein called calpain. It's an enzyme, and it has an active and an inactive form. And in this case, we know how these look like because they are in the PDB. But if they were not, then you would have no idea. And AlphaFold is actually predicting the inactive form. But again, you wouldn't know if you don't have it in the PDB, you don't know it's the inactive form. So that's something to keep in mind. It's not fixing all our problems. How you can use this? Of course, you can just take the code, the source code. I guess this crowd, you could just do it. You probably are quite tech savvy, so you could take the code and see if you can get it to run. Obviously, that gives you the most amount of control. You can also use interactive notebooks like Python notebooks, Google Colab. That's also quite good. It does most of the things that you might want, but obviously you can't really configure it as much. And then there's a database as well you can use, for example, the AlphaFold protein structure database, where you could get millions of structures. And this is the database if you haven't seen it before, but I don't really want to spend time on that. Instead, I want to highlight another thing, which is called the 3D beacons network. And the reason why I don't want to dwell on the AlphaFold database is because 3D beacons is expanding the amount of data and the sources you can get the data from. So the idea here is that often as a user or as a researcher, you don't really want to know or keep track of the different data resources who have structures for you. If you just want to work with specific protein or set of proteins, you don't really have to know or want to know, where can I get this data from? Because there are, I don't know, 50, 20 different data resources who have some kind of models. And so instead what we wanted to do, and we kind of stole the idea from the genomics research people who have a network as well, a beacons network. So here basically we have a unified programmatic access mechanism. And this is programmatic access. The point is not having a nice user interface. This is for programmatically getting the data. And so the idea is that you use these endpoints that we provide, and the endpoint will find out who has models for you, and then gets you the metadata and gets you the pointers to the actual files without you having to go and search in different places. There are quite a few providers in there already. AlphaFold database is there, but there are also others like the Model Archive, a Protein Ensemble database, and others as well. And there are several types of endpoints that you can use. I guess the most important, two most important are, one is keyed on UniProt accession. So that means that every protein in the UniProt database, it has a unique ID. And you can use that ID to get all the structures that anyone has who are part of this network. And the other thing you can do is to do a sequence-based search. So you just want to see what is similar to the sequence that you're interested in. Briefly, how this network works: we got together and still keep getting together with all the data providers who are members of this, and come up with schemas, mostly JSON specifications, to define how the data should look like. So these are kind of like data modeling, but in a collaborative way. And then we also create small lightweight Python packages, which are the beacons, the beacon clients. So people, the data providers, they set up these beacons, and then we have a hub, a beacon hub, that basically that's the one that people interact with. So if you come and have a protein of interest, you send a request to the hub API, and then the hub API pings all the different beacons and sees if they have data for it, gets back the response, collates it, and then presents it to the user in a standardized format. And basically that's how this system works. Like I said, the main purpose of this is the API, and this is for people who write code. But for demonstrative purposes, we did create a UI as well, just so that people can see what the data is, what types of data we have there, where you can get the data, and also we have a 3D visualization as well. That's quite handy if you just want to get an idea how this protein looks like. And one of the things that I already very briefly mentioned as an example is called AlphaFill. They are also members of the 3D Beacons network. And basically what they do is they kind of remedy this limitation of AlphaFold, that AlphaFold cannot plug in small molecules, even though when it actually would make perfect sense to put it there. Because, for example, it's a zinc finger protein. We know there is a zinc molecule there. And strangely enough, AlphaFold structures, even the side chains are in the correct orientation in many cases, as if anticipating the ligand to be there, but it's not putting it there. And so what AlphaField does, they plug in these small molecules that we know are supposed to be there. So they are more like completing the structure with small molecules. However, I think it's very important to know that this is not fixing all our problems. That on one hand, you have all these limitations that I mentioned. Often you don't really know what's going on, what conformation is this, and there are other problems as well, which I will try to show here. So fundamentally, if you look at the structures, these are just x, y, z coordinates of atoms, and it's just, you know, it's a cloud of atoms. On its own, it's not going to give you too much information. You really need to put it in some kind of context. And then by convention, of course, we have things like these ribbons. You know, okay, it's a helical part, or maybe it's a sheet. You can plug in the ligand as well, but you need to keep adding more and more information. You need to know that certain residues, there can be mutations there, and some disease are associated with these mutations. You need to know that these are the pockets where you have the ligands. You need to know many things about the certain protein and put these annotations on top of the structure if you really want to use them. And that's what the second half of the presentation is going to talk about, because that's exactly what we are trying to do in the PDB knowledge base. Here, this is a collaboration, and it's an open collaboration of different data providers and research groups who work with structures and try to provide pieces of this biological or biophysical context to really make these structures much more usable. It's an international collaboration. To date, we have about 34 data resources from many different countries, and it's really linking together structural bioinformatics, structural biology, and other research groups as well. The infrastructure itself is hosted and developed by the PDBe team, which is at MBI in Kingston. So this thing, PDBe-KB, I think we established it late 2018. And then the first actual user-facing thing was the aggregated views of proteins, where the idea was that if you are interested in a certain protein, you might want to see all the structure data for it. Because traditionally in the PDB, you have PDB entries, and they correspond to an experiment. That's not really what you are interested in most cases. What you want to know is protein A, how does it look like? What molecules bound to it? What's going on? So that's why we created the aggregated views, where you see all the data from across the PDB. And then there were other different things that we did. The 3D beacons, which is also part of the KB umbrella or under it, was released 2 years ago now. In terms of the data that we have there, we have tons of data. And if you are into data processing and data mining, it's really quite a valuable resource because we have many types of annotations. Mostly these are either functional or biophysical annotations, things like a ligand binding site, post-translational modification sites, or conservation, sequence conservation, mutations, the effects of mutations, different types of data. Some of the providers, they cover almost the complete PDB archive, and some are focused on very specific data sets. In terms of accessing the data and then using it, mainly programmatically, there are different ways. One is you can get the whole thing from the FTP area of PDB, and this means that the data, the annotations, they are in JSON files, but we also have these MMCIF files that I briefly mentioned. So these can include not just the coordinates, but also all kinds of annotations. And we have also updated files for small molecules. So if you are interested in solving problems related to drug design, drug discovery, then those are very useful things. Another thing that you can do is to take the whole database itself. And we provide a weekly updated database dump, really, that you can just download, set it up, and then use it as a data mining tool. That's actually quite powerful. And then in addition to this, we have the API endpoints. I would say maybe about 100 different API endpoints that serve all the data and data types that we have, plus the web pages, which are also quite handy. So one of the things I wanted to highlight here is the graph database, because the data we have and the way the data is interconnected, it was quite a natural fit to display it or represent it as a graph. And the reason why it's good for us, and many things, it allowed us to do many things, is because you can match very sophisticated subgraphs to this graph that can answer very specific questions. And running these kind of things in a relational database, that would often be very inefficient. But using the graph database, you can answer quite advanced questions. So here's an example, and this is quite a simple example, to be honest. You can get much more detailed. But suppose you want to see all the residues, so all the amino acids across the whole PDB archive that we see to interact with a cofactor molecule. And doing that is like super simple. It's much simpler than an SQL query would be. And it runs quick, and it gets you all the results from this graph. And then you can keep adding information to it, like it has to be a disordered residue, it has to be in a druggable pocket, all kinds of things you can add to it. And in that sense, it's a very powerful research tool. If you want to access this database, it's absolutely free. You can go to that page and you have a download link. Well, the only caveat is that it's kind of big. It's about 0.5 terabyte, the database itself. And we have, I think, about 2 billion nodes. So it's not a small database, but I think it's worth it. And we also provide an interactive schema that describes the data and really helps you to construct the different queries that you might be interested in. And I think in the last part of the presentation, I just want to give a few examples of things we are doing in the protein data database knowledge, data bank knowledge base. One of them is to do with distinct conformations of proteins. So the same protein, it might have different conformations, and it's kind of hard to find these in the PDB. They are not really annotated. It's not easy if you want to see an active or inactive form, or open, closed, and so on. That's one of the things we were working on, to create a process to help people with this. Another thing that we are looking into is in terms of data visualization and 3D data visualization primarily, where we want to create a viewer, a 3D viewer, that can go across the scales. So it can go from atomic view all the way to basically microscopy images. And the third thing I wanted to highlight here is a process that we had to create for complexes, because in the PDB, everything that has more than one component, a macromolecular component, that's a complex. And the problem in the PDB is that these complexes, the unique compositions, are not really recorded, and there are no persistent IDs. So if you are interested in a particular complex, in a particular composition, but across the whole PDB, it's very hard to get that data. And so to fix that, we had to create a process that can uniquely identify complexes. So the first thing is, and this is the work of Joseph Allaway, who is the main developer for this pipeline. The problem here is that X-ray crystallography is really good at determining single conformation, and most of the entries in the PDB, they come from X-ray. But proteins are often dynamic, and they are more on a scale. So on one hand, you could have completely rigid structures, but I would say that that's completely rigid, that's quite rare. And then you have very, very flexible things like these intrinsically disordered proteins that just, you know, float around and can have all kinds of conformations. Most of the things, they are between or at some point of the scale. Until recently, it was very hard, especially if you didn't really work with these types of structures before, to find the distinct conformations for a protein that you're interested in. And we just wanted to create a process that does this automatically for every new entry as well, so that people don't have to bother. It built on something that we already had for a couple of years, which was to superpose all the structures. So basically, you have the structure and you try to overlap them or overlay them. And what we wanted to do is take all these superposed structures and cluster them based on structural similarity, so that you can come up with distinct conformations. And then ideally to assign a biological relevance. Why is it looking like that? Is it an open state or closed state, active, inactive? Why does it look different? Another thing that we wanted to do here is to superpose ligand binding sites, which means that, for example, you know that there's a certain drug molecule that binds to a certain binding pocket in one protein, but you are interested maybe if you can target any other proteins with that drug molecule. And then if you actually have a convenient way of superposing all the binding sites that bind with this ligand, then you will have a very quick way of knowing, oh, actually, I could maybe target another drug as well with this particular drug molecule, not drug target. So this started quite recently, so last year, and it's together with the CCP4 community. And we also work on a tool called Gesamt together with CCP4 to make that better for our use cases. We already have some preliminary results of this, and if you would go to one of these aggregated views of proteins pages that we have, you can get access to all these superposed views. And on one hand, you see these clusters, but if you look at the right-hand side, we also show the AlphaFold structure. So you can superpose the AlphaFold structure and see which conformation does it resemble. For this, we had to make some changes. We have a 3D viewer called MoStar, and that's an open source tool. I think it's WebGL based. That you can take. Many data providers use that tool. This is together with RCSB PDB as well, so they also use this MoStar viewer. And we had to add some support for the MoStar for the AlphaFold superposition as well. One thing I want to highlight here is that we always show the PAE plot. So whenever we display a predicted structure somewhere, we put this PAE plot next to it because it's very important to understand what it looks like. And here's another example I wanted to show also in the context of AlphaFold. This hexokinase, it has different conformation depending on does it bind a sugar molecule. And it turns out that in this case, AlphaFold is predicting the unbound form. So when it's lacking the sugar. But again, you can never know. In other cases, it actually predicts a structure that should already have the ligand. So it's it makes it a bit complicated. And in terms of how we worked on this, Joseph was creating manually curated training data set and did some data cleaning. And then basically the clustering itself, which will be described in detail very soon. I think next week we aim to put it on bioRxiv. So basically, it's not using the superposition; it's looking at C alpha distances, and based on that and some custom formula, it can come up with the clusters. The difficulty there was really deciding, because I think we are using hierarchical clustering, and the question was where to cut the tree and where to be able to say that these are the distinct clusters. But yeah, so that was the challenging part there. Another thing that I briefly mentioned is how we can visualize data at different scales, because of course, in terms of molecular structures and molecular data, we can go from atomic level through these low resolution envelopes that kind of like give you the overall shape of something, all the way to cells, whole cells, tomography and microscopy images. So it would be quite neat if there would be a tool that you can use and go across the scale, all within an integrated environment. And this is roughly what I mean. So basically starting from an atomic resolution structure, going all the way up as far as the data actually lets you. And the way to do this, we initiated a project recently, and this is together with RCSB and SciTech, which is in Czech Republic. And the goal here is to take the MoStar viewer and expand it so that it will be able to support multi-scale data visualization. And not just that, but we also want to be able to compare and contrast the different levels. So on one level, to be able to contrast and superpose and describe what are the differences, and to map annotations on each of these different levels of data. The way it would work in terms of the technical details, on one hand, we have data providers like the PDB or the EMDB bioimage archive, and we would provide some kind of data layer for them that kind of transfers the data, transforms it, and then links it to an API, where the API would be there to integrate and then deliver all this information in a standardized format to CellStar, which will be the actual front-end viewer for all this. So that's something that we recently started. But another thing that we actually already finished is this work on the complexes. And there, as I mentioned, the problem is that the PDB curation process actually doesn't require a depositor to say or uniquely identify a complex. And because of that, in the PDB, it's very hard to find a specific complex we are looking for. Even if there is a well-defined name for that particular complex, and that's used in literature, even with that, you would need to rely on text search, which is obviously not very optimal. And in other words, complexes are just not findable enough in the PDB. And to remedy this or to fix this, what we needed are persistent and unique complex identifiers. The way to do that, we were creating a pipeline that can look at the composition of all the different complexes, and then based on the composition, we could create unique hashes, which we can then turn into unique identifiers that now we integrate into our databases and then use it downstream. So we can use that in our APIs, we can use it on our front end. And eventually what we want to do is to aggregate all the structure data for a particular complex. So maybe you work with ribosomes and you want to see all the structural data for a certain ribosome, then this would be a good way of getting that information. Maybe it's important to know that this is together with Complex Portal team, so we are coordinating with them. That's a data resource who specialize in complexes, but the problem is that they have a tiny data set compared to the PDB because they are painstakingly curating it by hand, and so their data set is much smaller. In terms of practice, what it means is that now if you have a complex that you're interested in, you can either get mappings for it from the FTP area. So on there we have CSV files that have all the information about all the complexes we have in the PDB, or you can use the search system, and there you can click on things like this, search this complex or search this ID, and then you will see all that part, all of those instances across the complete PDB archive. These are just a few of the things that we were doing. And if you're interested in seeing what else we are trying to do, then please visit the page that we have, so PDBe-KB.org. But I thought these are maybe interesting highlights. And to repeat my main messages, even though we have quite a few entries in the PDB, and we have 800 million structures in different databases that are predicted structures, and these are very nice tools if you want to well, for drug discovery, for disease mechanism understanding, for structure determination, all kinds of things. But still, you have to really keep in mind two things. One is that always look at the confidence metrics, especially if you are working with predicted models, but actually even for experimental structures, always look at the quality. And predicted or not, you still need to have some kind of context for it. And that's what PDBe-KB is trying to do, to put these structures in their context. And so before wrapping up, I just wanted to show this slide because it has a couple of useful resources. The FTP area, where you can get all types of files and data that we have, annotations, benchmark data sets, various things. Then the graph database, as I mentioned, you can grab it from there. And using the schema, you can use it as a data mining or research tool. We have documentation, quite extensive documentation for the API endpoints that we have. And of course, we have a couple of pages, web pages that you can use. And clearly, it kind of takes a village to create these data resources. So we have a lot of people, and I would like to acknowledge all the work that they are putting into this. And of course, acknowledge all the funders that are helping us. Thank you for your attention.
Thank you, Mihaly. Okay, so I'll stop the recording. And